The aim of my project is to develop and validate an organ-on-a-chip model of the choroid plexus, the part of the brain that secretes cerebrospinal fluid. My long-term goal is to treat hydrocephalus, a neurological condition characterized by the buildup of cerebrospinal fluid in the ventricles of the brain, causing an increase in pressure inside the skull.
We're proposing an alternative stem cell-based model to replace many of these animal models in neurotoxicity testing. Thankfully, the lab I work with, the Centers for Alternatives to Animal Testing, already have their own in-house 3D human stem cell-based model in order to model brain organoid systems. As such, I've been able to take that model and add onto it and innovate in order to fit the needs that we have for our neurotoxicity endpoints. We can take various measurements over several weeks to explore and analyze the effects these chemicals might have on a developing brain and its neurons.
As the title suggests, in this project I am trying to advance in vitro, so non-animal models of kidney tissue to make them more suitable for testing drugs in the preclinical pipeline. If successful, we will be able to use what we learn from these new models, not only for safety profiling, but also for drug discovery, basic understanding of human kidney development, and ultimately engineering larger scale tissues for therapeutic purposes.
My PhD project is to engineer a organ-on-chip system that mimics the function and structure of the human kidney glomerulus. The successful development of our organ-on-chip system will allow the researchers to study kidney disease in a more physiologically relevant way, instead of using animal models that have a lot of ethical concerns.
The aim of my research is to use brain tumor organoids to evaluate the efficacy of novel inhibitors. One such model is patient-derived organoids, which display similar characteristics and growth rates to those of human brain tumors within patients. However, these three-dimensional cell structures are not readily used, despite being superior to current two-dimensional models in their ability to mirror features of human brain tumors in patients.
My PhD research aims at understanding the molecular mechanisms that underlie the liver's regenerative capacity.
The approach we're taking here at our lab is to make use of a newly developed culture system for primary human liver cells, which come from organ donors. This culture system relies on having a 3D structure which allows these cells to display their true biological characteristics.
The ultimate goal of this work is to generate knowledge that will be relevant for the development of future therapeutical strategies to treat and eventually cure liver diseases, which are becoming a big burden in the clinics nowadays.
My project particularly aims to assess the developmental neurotoxic potential of a class of man-made chemicals called as flame retardants. In this project, I will be assessing the developmental neurotoxic potential of these chemicals using a stem cell-based 3D in vitro brain spheres. These models for chemical testing have the potential to reduce, refine, and replace the currently used models for chemical testing.